Welcome to the Masters of Beautiful Achievements podcast, where we share the insights from inspiring masters about how the use of physics, biology, and green chemistry is shaping our future. It's all you need to know about innovation and better business models. The podcast is hosted by Alexander Prince, founder of Scope Matters. For more inspiring episodes, visit the Master of Beautiful Achievements website. Today's episode is part of a special series where we look at microbes through a different lens. We will explore how life sciences, biotechnology, and environmental sciences are influencing how we look at our natural world. During this series, Alexander Princeton and Mark van Bemmel are curious to know what happens when we zoom out and look at microbes as important players in a much broader system. How does this different perspective influences on how we look at the role of microbes and viruses in the web of life? And what lessons can we draw from nature's logic into making our world more sustainable? Together with specialists in the field of virology, microbiology, and biochemistry, we go on a journey combining insights from various disciplines to learn about the essential questions we might have been overlooking. And who knows, we might discover one or two innovative applications along the way. This series is brought to you by Scope Matters, a systemic innovation agency from Rotterdam, the Netherlands. They accelerate business leaders through systemic innovation by merging ecological thinking with economic doing. Their system approach transforms and improves the business model of their clients by creating new opportunities, mitigating risks, and increasing cash flows, while at the same time eliminating waste and doing good. It is the space where systemic sustainable thinking meets circular innovation, because in the end all matter counts. Check out the website on www.scopematters.com. And Orvion, a young biotech company based in Stolwijk, the Netherlands. They study microorganisms and are developing their own DNA analyzing techniques, like for instance the Orvion U-Detect. They work on solutions to remove contaminants from ground, water or industrial processes more cheaply and permanently. Their services are available for water treatment, soil remediation, ecology and industry. Orvion believes nature can do a much better job. So please take a look on their website on www.orvion.nl. Welcome to this virtual Venture Cafe session in Rotterdam. Today, Mark and myself are hosting this monthly session. The theme is Microorganisms Through a Different Lens. It's a podcast series by the Masters of Beautiful Achievements and hosted by Alexander Princeton, who is founder and managing partner of Scope Matters, and Mark van Bemmel, who is founder and CEO of Orvion. These are a series of explorative dialogues where we look at our natural world from different perspectives, and along the way we will discover how it impacts how we look at life sciences, biotechnology, and environmental sciences. For each session, we will invite a guest speaker, a specialist in his field of virology, microbiology, or biochemistry. And for today's sessions, we will explore and investigate how, and especially also why, microbes are able to crack chemicals like PFAS, and how they found ways to bind metals, of which normal scientists would say that was impossible. My name is Alexander Princeton. With my company Scope Matters, we help companies to accelerate their innovation capacity by merging ecological thinking with economic doing. And we apply natural science and systems thinking to make this happen. For today, our special guest is Peter Jaffe from Princeton University. Peter, please have a short bio about yourself.
Hi, thank you. Yes, I'm Peter Jaffe. I'm in the Department of Civil and Environmental Engineering at Princeton University. I work in biogeochemical cycles affecting water quality, looking both at water treatment, wastewater treatment, but also cycles and how it affects specific water quality parameters at the local scale, meaning contaminated groundwater or wetlands. And we have done even work at the watershed scale, looking at nitrogen cycling and how changes in climate or climate variability may affect nitrogen cycling at the large scale. I've been at Princeton on the faculty since 1985 and enjoy moving from different topics in this sort of fascinating field of fate and transfer of contaminants and how the physical and biological processes interact.
Perfect, thank you. And Mark, could you share a little bit about your background?
Yeah. I'm Mark van Bemmel. I'm founder of and CEO of Orvion. At Orvion, we look at bacteria in water and in soil with DNA techniques. So we try to establish who is there and what he is doing. And by doing that, we try to make the world a little bit cleaner and more sustainable. And I would like to keep it at this short introduction. So.
Because we need the time to talk to Peter
Right.
To learn about all his insights. Peter, I went through one of your lectures online, and I have to say, I learned something new, which was, I've never heard about that, but it was the Fearmux bacterium.
Fearmux, yes.
Fearmux bacterium pathway.
Yes.
And I understood that it's quite a breakthrough to understand how that actually is working.
Yes, I can talk a little bit more about it. This was something we discovered, and I have to tell my students, keep your eyes open for the unexpected. We were doing some field work in the early 2000s, and we were actually looking at the runoff of agricultural nitrogen into wetlands and how that affects the iron cycle. And when we were doing our field work, we had a terrible drought, and so our wetland was dry, and so out of desperation we took core samples into the lab and incubated them there. And there we noticed that ammonium was decreasing in this anaerobic incubations. Nitrite was appearing, and but we were under iron reducing conditions, and there was no initial nitrate, so it couldn't have been the Anaerobic process by which ammonium can be oxidized and so we postulated that there is this process by which ammonium can be oxidized under iron reducing conditions. Just a year later, this a Japanese fellow, Shawayama, I think was the name, reported something similar in a reactor, and he called the process Fearmux as a play to Anaerobic. That paper is very lacks detail, so but the contribution of that paper was mainly to come up with that name, Fearmux. A little bit later, a group from Berkeley reported something similar in rainforest soils in Puerto Rico. And then we went back and got more samples from the site. I hired a microbiologist then to help us identify the process. And we during a series of incubations, we realized that there was this novel organism, which we called an Acidobacterium A6, and the A6 was because of the DGGE lines, the sixth band. It was of group A, and that's not much imagination. We could have done something more fun with coming up with that name, but before I could come up with something more fun, and my postdoc, Dr. Huang, had already submitted the name, so it stuck. So that is a new, at that point, well, there are lots of unidentified bacterium. So this was a new Acidobacterium, and we have since been able to isolate it and actually show that it can do this Fearmux process, that it can oxidize ammonium under iron reducing conditions. And you look at the energetics, and it is something that should happen only under acidic conditions. It becomes unfavorable under alkaline conditions. We have then done, as soon as we published that, back off, we were approached by the Chinese Ministry of the Environment. For them, ammonium was in their 5-year plan one of their top priorities, and they wanted to collaborate with us. So they sent us a lot of samples from Southeast China. We had to go through the whole protocol of importing soils with the US Department of Agriculture, etc. But we took a lot of samples here in the East Coast and then from Southeast China. And we realized then that this organism and this Fearmux process is actually quite common.
Oh, wow.
We found it typically in wet soils that are acidic and iron rich.
But that means that these are also soils which tend to be hard for plants to grow on.
Not necessarily, no. I mean, in some cases, they're wetlands. We actually have more recently also found that organism in river sediments.
Okay.
But wet soils, I mean, it's a little bit like there are many organisms that don't like the presence of oxygen. And let's say many of the denitrifying organisms, you find them in soil when you have water, when the water blocks the diffusion of oxygen. And so you have micro locations that are anoxic, and then you have denitrification. Something similar with that organism. We don't need deep reducing conditions. We just need the water to block the diffusion of oxygen. There shouldn't be nitrates, but once the nitrates are depleted, we get into iron reducing conditions. And
what are iron reducing conditions? I know a lot about biochemistry and microbes, but there is so much more I don't know.
So let's go back. Organisms usually to get energy from a reaction, they have usually something is oxidized and something is reduced, and that reaction frees energy. The typical compounds that get reduced, or the reductant would be, and the most typical is oxygen, right?
Yeah.
That gives you the most energy. If you, let's say, use organic glucose and oxidize it with oxygen, you get the most energy, and that's why aerobic organisms can grow the fastest. But once you have no more oxygen, there's a series of other reductants that different bacteria can use. The next energetically more favorable after oxygen would be nitrate.
Yeah.
And then very typicals would be manganese, and the next one would be iron. Then comes sulfate. So what iron reduction means is that we have compound like organic carbon that is oxidized and transfers the electron to ferric iron, iron +3, right? Converts it to ferrous iron, iron 2. And there's a significant amount of carbon that is turned over under these iron reducing conditions. And there are lots of heterotrophic iron reducers that, like I say, use organic carbon as the electron donor. But the organism that we are working with, this Fearmux bacterium, is an autotrophic organism. It doesn't use organic carbon. It uses the ammonium as the energy source and oxidizes the ammonium to nitrite. And the electron gets then transferred to ferric iron, which is reduced to ferrous iron. In the process, it gets energy to grow. So we're talking about an
interesting
anaerobic autotrophic iron reducer.
Yeah, they have the TU Eindhoven has now a technology where they can use iron oxidation and deoxidation to produce energy, which is like you're just describing it from a biological point of view, and the guys have figured it out from a physics and a chemistry point of view. Interesting. Wow.
You can almost say, or you can venture to say that if there is a reaction in the environment that can produce energy, some organism will have made a niche and is managing to make a living from getting that energy.
Yeah, Peter, I read your paper on these microbial cells that where you can do this Fearmux process in.
Yes.
And then actually, if I understand correctly, the ammonium is oxidized anaerobically, so you don't need oxygen.
Right.
And you produce some electricity in the going. Is that right?
Okay, so that's okay. Let me get this. So and there's some interesting we're oxidizing anaerobically the ammonium to nitrate, and so the big question, and I don't have an answer, is where is the oxygen coming from, right?
Right. Right.
And I'm working with chemists, enzymologists from our chemistry department trying to figure it out. There is a gene for an enzyme that looks like an oxygenase. Oxygenase is typically enzymes that insert oxygen, and there are ammonium oxygenases. So this one has a lot of similarity to a methane monooxygenase, which is the enzyme that puts oxygen and methane, converting it to methanol, which is the first step in oxidizing methane. But where's the oxygen coming from? So our chemists have a whole bunch of ideas where the oxygen may be coming from. We haven't even dared publishing that in the academic literature because people would want to know where the oxygen is coming from, and I don't know. No?
Is it coming from water maybe, or
well,
it could be. You can electrolyze water.
Bacteria.
Not
impossible, but there are not this many organisms that split water, right? Photosynthesis does, so it's not. It could come from water. It could come from the oxide, from the iron oxide. We don't know.
But the interesting thing is that you can, in this microbial cell, you replace the iron 3 for electrodes. Is that right?
Yes.
Yeah.
Yes. So we want to grow this organism for multiple applications, and we have been able to grow an enrichment culture in a membrane reactor. But when you look at the stoichiometry, you need 6 moles of ferric iron per mole of ammonium. That's a lot of iron.
A lot of iron.
So,
yeah.
You add all of that ferrihydride to a reactor,
so,
and it clogs up after a few months of operation.
Yeah. You have to put in iron all the time.
Right.
Yeah.
And we tried, unfortunately, we tried. It doesn't mean it's not possible, but we could not find any dissolved ferric iron. I mean, there are some geobacteria so that can use ferric citrate also as the iron electron acceptor. This one we haven't been able to find any dissolved iron. Doesn't mean it doesn't exist. There could be a specific redox potential or pH at which it may work, but we haven't got there. So then there are many iron reducing bacteria that are called electrogenic. They can transfer, if you have a little anode and a cathode, they can transfer an electrode to the anode instead of iron, and then you can grow them without iron. So we decided to check if this bacterium does that, and yes, it can. So we start out by putting little metal plates into the soil, deep where it's more reduced, and shallow, and we could see that it would colonize and connect it with a wire, and that it would colonize the deep ones. If we had a parallel graphite plate without being connected to the more oxidized one, it would not colonize the graphite.
You see that?
Yeah.
So then you see this acidic microbiome grow on the anode.
Yes. So that was a paper that came out in Applied Environmental Microbiology in 2019.
Yeah.
And then we decided after that to grow them in these microbial electrolysis cells, which is the same thing but in a little vial. And you put an anode, a cathode. Now you don't have a natural redox potential, so you put an external redox potential in a little. We put in 0.3 millivolts with respect to the. It's always with respect to some kind of reference electrode, silver chloride reference electrode, and we could see that the organism would grow and oxidize ammonium.
Right. So then you have an anaerobic ammonium oxidizing system. So it doesn't.
Yes.
You don't need any energy input other than putting in the ammonia, is it?
Well, right, and a little bit of external power, but
very little.
That's minuscule. So
so then you can crack ammonia by just doing that.
We can oxidize ammonium by.
Yeah.
Well, yes. So we are in these little vials, and scaling that to an industrial process would be fantastic, but we're not there.
Yeah. If you phrase it this way, there are two things in my mind popping up: as a microbial fuel cell, and the second thing is if you are able to do that, are you able then to create fertilizers?
No, but our goal was more when you look at your traditional wastewater treatment plant, maybe 50% of the energy going into domestic wastewater treatment plant is to oxidize ammonium.
Okay.
They are, and as we move more and more towards trying to design anaerobic treatment plants so that they don't have this much of an energy input, and you can actually produce ammonium and make the plant run sort of energy neutral, we're still stuck with needing to oxidize the ammonium. There is a reasonable progress with implementing Anamox for anaerobic oxidation, but it's not truly anaerobic. You do need aerobic oxidation. Partial, half of the ammonium would have to be oxidized to nitrite.
Yeah.
And then stop it at
put in a
nitrite.
bit of oxygen.
Yeah. And so there are some plants that run successfully.
Maybe can I ask you one question, Peter, about this? Actually, this Anamox has been evolving all the time, which is a very nice biological way of treating these wastewaters. But you need quite a high temperature for these Anamox bacteria. It doesn't really work under low temperatures. Is it the same with the Fearmux process? Is it all?
No, we have seen that it actually works quite well at ambient.
Yeah.
And we didn't do the full gradient of temperatures, but we did 10, 15, 20, 30, and 20 was by far the highest rate. So that's an advantage. It would work better at ambient.
But I mean, Anamox works really nice at 30 degrees, but
yes.
Below 20 is really hard. Yeah.
Right.
That Fearmux could be more efficient at lower temperatures.
If we can make it work, yes.
Yeah.
So we have a continuous flow reactor, and we're struggling with it. There's something not quite right. So it works for a little while, but then it dies on us. So we have to figure out and I tell folks, and the university has some patents on it, and says, look, I don't know if we're 2 weeks or 2 years or 20 years away.
You never know, is it? How do you keep going then, Peter?
Well, it's because it's fascinating.
Yeah, that's what it is. It's fascinating. And it gets even more fascinating, I think, because you also found that this acidimicrobium, by oxidizing the ammonium and reducing the iron, also can put its electrons on PFAS and thereby destroying the PFAS which is
it can transfer the electrons to many different compounds. The first one we looked at, because we have done maybe a decade ago a lot of work with the Department of Energy on immobilizing uranium using bacteria, and you use this iron reducing bacteria. Uranium is in the environment sort of two typical oxidation states. +6 is very water soluble, and uranium +4 is insoluble. So you can use this iron reducing bacteria. If there is uranium-6 present, they will transfer electrons to the uranium-6, convert it to uranium-4, which precipitates out as a uraninite solid. So we checked with this Acinetobacterium A6, and it can reduce uranium-6.
Of course.
So that was fun, because it links, then we can do it with ammonium as electron donor as opposed to CARM. Interesting.
Interesting.
Probably CARM is more easy. We could show that we could reduce copper-2 to copper-1.
Also.
Yeah, it's not.
So it doesn't really matter which metal it gets, is it?
No, I mean, these are all secondary, so we need plenty of iron, right? Because they need to be there in the millimolar level, and if we were to put uranium or copper in the millimolar level, it's toxic. So, but again, copper-1, I don't know what to do with copper-1. It's not terribly useful, but it's just as a matter of showing that it can transfer electrons to different metals or radionuclides. It was interesting. Then we noticed that once we had isolated the organism and we sent it out for genomic sequencing, that it has several dehalogenases. And we had done some work originally that showed that we could degrade trichloroethylene. We did that because it has this enzyme that is similar to methane monooxygenase, and we know that methane monooxygenase can epoxidize trichloroethylene. So we showed that, and we never saw the production of dichloroethylene, so we thought it was not a dechlorination, it was probably an epoxidation. We may need to go back and revisit that, but then a colleague, former postdoc that is working at a consulting firm says, can you degrade DBE, dibromoethylene? And I said, oxygenase should not do that, but he says, why don't you try it? We do the analysis, so we tried it. And there was not much, but over a month of incubation, there was a statistical decrease of 10% while the autoclave control stayed constant. And that bugged us because the oxygenase should not have done that. And then looking in detail at the...
So it must have been a reduction. That's what you said.
Yes.
Yeah.
We did not look at bromide or anything like that to see. But,
just a simple test. Yeah.
So then looking at the genome more carefully, we realized that the organism has a few dehalogenases. And that decided, given the interest in PFAS, I mean, under normal circumstances, I wouldn't have started with PFAS, but given the interest, I said, let's for the moon.
That's just a wild idea, actually.
Right. So I thought it's a long shot, but fine. So we bought. Yeah. And it was also driven because EPA had a call for proposals, and we said, we had some initial results. So we bought 20 different PFAS, and we did quick, dirty incubations. We checked first toxicity, made sure we were below toxicity ranges. Mm-hmm. And 2-3 weeks later, my postdoc, Dr. Huang, came to me and she tells me, I see fluoride production. And I said, in which one? And she says, in all of them. that is just cannot be. No? So
you must have been very happy that day.
That was well intrigued, right? So,
yes,
first when you expect something unexpected, you give the poor person grief and check where did you go wrong, what is wrong.
You didn't do your measurements right. So.
Right. But then we did it very carefully over, and we focused on PFOA and PFOS, which are the two of PFAS of most regulation, most concerned, and most common.
In Holland, by the way, Peter. Yeah, we have a lot of it around us here.
Yes. And that the results came out quite interesting. We were sitting on it for a long time. We did send everything to, we don't do our own PFAS analysis, so we send it to multiple labs for verification. Fluoride analysis are not easy, so we did them, we do them mostly with ion chromatography, so we did them with 2 different columns, different elements, over and over. I kept telling the poor Dr. Huang, do it over, do it this way, just to be convinced. And then we still sent it out, and we didn't have at that point a fluoride electrode, so we sent it out to be analyzed with a fluoride. Once everything came out consistent, and we also noticed the degradation products. We started with PFOA, and it wasn't only that was going down, but we started to see perfluoroheptanoic acid, perfluoropentanoic acid, butanoic acid, all of them appearing as the perfluorooctanoic acid decreased. So we said it's real, and again, it's sort of, we aimed first for one of the big Nature communications or so, and then another one, the Proceedings of the Academy of Science. Both times the editors told us this is not an area of broad enough interest.
Really?
They didn't send out for review. They said,
Whoa, such a
Oh,
big
wow.
finding, and then
I
no,
know, I
no
know. And so
interest.
I was shell shocked, and
That's horrible.
I
Why?
They were
because
scared.
they were not informed properly or scared. I don't know. I don't
I
know.
mean,
So
maybe to clarify to our listeners, PFAS are some of the most toxic compounds ever made by human beings, isn't it? They're extremely toxic, and even in very low concentrations.
well, I don't. They're toxic. I'm not sure if they're the most toxic. I mean, they're really not meant like pesticides to kill something.
No, no.
But they are among the most stable compounds, right? And they do have health effects. And because they're so stable, and because they have been used in so many products, there's virtually, at least in the US, they say that, I forgot the exact number, 98% of the population has PFAS in the blood serum. It's probably
Oh.
similar worldwide. In Europe, probably very, very similar. They are linked to multiple health problems, including some cancer forms. The best study was one. There was a movie last year, Black Waters or Dark
Dark
Waters?
waters.
Black
Dark
Waters.
waters. Dark waters. Yeah.
Dark Waters. Dark Waters. That, as they show in that movie, DuPont agreed to a very, very large a medical study, and it was sort of a natural control. There was this reasonably large population that had been exposed to PFAS, and they could document a series of health effects. Anyway, we were quite amazed that this organism can defluorinate them. We have shown that it can defluorinate many other compounds. I won't give much away. We're now working on a paper. We have shown that it can defluorinate PFAS in these microbial electrolysis cells in those reactors. So it
I wanted to ask that question.
So we're working on that paper, but it opens a pathway now to maybe have a reactor that we can use that.
Yeah.
We have preliminary results showing that we can defluorinate PFAS in biosolids from wastewater treatment plants.
Mm-hmm.
We need to seed it with that organism and incubate them anaerobically. PFAS in biosolids is a big problem that I hadn't realized until we had that paper, and I got a lot of phone calls from operators of treatment plants. Right now, and I imagine Europe is similar in the US, that in biosolids, which is for those that are not familiar, in a domestic wastewater treatment plant, you convert the organic carbon. Half of it goes out as CO2, half ends up in bacteria biomass, and that biomass gets digested a little bit, but still you end up with a big plant, several tons per week of bacteria. They're fantastic nutrient, carbon, nitrogen, phosphorus, and they're applied to agricultural lands. But if they have PFAS, it may be much more challenging to apply them to agricultural lands. And so many operators of treatment plants are very concerned and wondering how can they treat these solids, because the PFAS just move through the treatment plant without being degraded. So we have shown that we can augment solids with this organism and degrade them there. So it's all very exciting. The only problem is we deal with this organism that is an anaerobic autotroph. Autotrophs grow slowly, anaerobes grow slowly, right? So if you want it in perspective,
the panda bear, or how do you say?
Yeah, the panda bear of the bacteria.
Yeah.
E. coli has a doubling time of 20 minutes, right? This one is somewhere between 10 days and 2 weeks. But
it has to do a very thorough job.
Yes.
To chuck off the fluoride. So he's I don't know. He needs the time to do so.
Yes.
I'm just curious because while you guys are talking, I'm also checking up on the internet. Is PFAS similar or related to organofluoronize?
Yeah, PFAS stands for per and polyfluorinated alkyl substances. So
Yeah.
there are about probably somewhere 4,000 fluorinated organics that have been synthesized.
Because it is also it does occur naturally, or microorganisms do naturally create organofluoronize as a part of their metabolic process.
There are some natural, look, there are lots of natural halogenated compounds.
Yeah.
And also some natural halogenated fluorinated compounds. I have a colleague in our geoscience department that studies them. But they are typically not as heavily fluorinated as this, this human synthesized one. The reason that they're interesting is that the carbon-fluorine bond is the strongest covalent bond in organic chemistry. And, and by having such a strong bond, and especially if the compound is fully fluorinated, like PFOA, where all carbons are fluorinated except the functional group, the carboxylic group, everything else, there's no carbon hydrogen, just carbon-fluorine bond. Those are so stable that they can resist very, very high temperatures, right? So that's why you can make your Teflon. Teflon is like carbon fluorine. It's you used to use the perfluorooctanoic acid and polymerize it, so to make Teflon. That's why you can have a Teflon in a pan and heat it to very high temperatures, and it stays stable. Firefighting foam, again, many PFAS are surface active surfactants. So firefighting foam is a surfactant. It makes bubbles, but by being fully fluorinated, it's stable under high temperature. That's why you use this firefighting foam and can put it on oil fires so that are very hot, but it doesn't burn. It just asphyxiates the flame. So it has many useful properties, but by having such a strong bond, it's also hard for bacteria to degrade them.
Yeah, and that's why they need a lot of time to break it down in a way. Interesting. I mean, it's good to know that in nature the bond is naturally occurring. Only humans have extrapolated that into a structure that is very unnatural.
But Peter, when we about 5 years ago, the PFAS problem in Holland became much clearer, and we started as a company. We started asking around for funding to look at biodegradation of these compounds, but actually nobody believed at all that they could be degraded. Have you met such skepticism also, or?
Yes, and there are. Look, I think our results are very, very convincing, but there are some people that are somewhat skeptic still.
Still, right.
And that comes to a large degree because it was generally accepted that the PFAS are not degradable. And I have to be careful. PFAS, the polyfluorinated ones that are not fully fluorinated, those are degradable. There are bacteria that can convert a very large fluorinated compound in a smaller one, but at the end you always end up with what's called a perfluorinated part, right? So, and once you ended up with this perfluorinated compound, there was the general school of thought that they cannot be degraded any further.
And it was the same many years ago with tetrachloroethylene, for instance. People thought that because it was chlorinated all the way, it wouldn't be able to be degraded.
I think when you look 20, maybe longer, almost 30 some years back, people would say this something very similar with chlorinated compounds, PCBs, and so that they are not.
Now it's an established technology to use bacteria to clean up soils, chlorinated solvents. Do you think the same will happen for fluorinated compounds?
I hope so. I think I mean, what we have discovered, and I just don't believe that this is the only organism. There has to be some others too. If there's one, there are more. That technologies will be developed for that purpose. We will be working on it.
Yeah.
We just have to be able. Look, there are two directions when you look at a contaminated site, right? We have obtained samples that were contaminated where the geochemistry is the right one for the organism to exist. Acidic, iron-rich, where we did some incubations and we could then do DNA extractions and show that the organism was there. So then we just incubated it. We added whatever may have been missing, ammonium, or in that case, ferric iron was missing. Most of the iron was ferrous. And we could see that we started to produce large amounts of fluoride. So there could be contaminated sites where the geochemistry is so that the organism exists but may not be very active. So then we can very simply manipulate the site and
get them to grow and work for us. Yes,
there are some challenges, right? And one we're working on is we need ferric iron, right? And typical in bioremediation, if there's an electron donor and an electron acceptor missing, we inject it. But ferric iron is a solid, so it's not quite as trivial to inject it.
I can imagine.
We have done work in coating ferrihydride in polymers and supply it that way. So we are working on that and having a new project starting to focus on that. So that would be one option. The other one would be is if the organism is not there, right? Then you do with chlorinated compounds what's called bioaugmentation.
Augmentation, right?
There are companies that grow this organism and then you inject a few cubic meter of this high density solution. We have to be able to get to grow that organism, and it has to be in these bioelectrochemical reactors, because we just cannot inject this slurry of ferrihydride and organism. And that's where we're still stuck a little bit, and like I said, something we're still missing.
But
there's one. Sorry.
Just curious, Mark, and I need to jump in. Could it then be that you're missing another organism because they're used to live in microbial flora systems?
Well, when we are doing both, we're working with a pure organism, and we are also working with enrichment cultures, and the enrichment cultures are much more stable, and it grows better in enrichment cultures. The pure culture we just do use more as a proof of concept. We want to show this is the organism that does this job, but we cannot 3 months or so and it disappears on us. So it needs the whole ecosystem to grow. So we constantly have to reculture it to maintain our pure culture.
Yeah, one question that bothers me a little bit, or would be fascinating to know why this happens, this defluorination, because apparently it's not because of toxicity or so that the bacteria wants to get rid of. It's
right. So look, we have a hypothesis, but it's just we haven't done much testing. We wrote a proposal.
Never mind. I like to know your ideas.
Anyway, so what we have shown, especially when we put these electrodes in the ground, and we have put a lot of electrodes over a fairly big area, then we come back and we scrape off and do DNA extraction and look for the whole bacterial community. What we typically see is a very strong negative correlation between bacterium and heterotrophic ion reducers, Geobacter, Shionella, and so it has a hard time competing for the surface to get rid of the electron with those organisms. possible idea would be that it has all these dehalogenases because there are so many natural halogenated compounds to help it get rid of the electron.
Yeah, it
when
needs
has
a
hard
bit
time
less
completing.
iron maybe than the other guys.
Yes,
Something like that.
That's the hypothesis. So we need to test it. And I've been talking with this colleague of mine that works with natural halogenated compounds. To
Or do you think it's even easier to get rid of the electron on the PFAS for this acidic microbiome than on the iron?
Well, I don't know if it's easy or not. There is a gene that is expressed when it's transferring the electron to the PFAS. So there is some extra work that the organism has to do for that.
Right, exactly.
And again, not being an enzymologist, I passed that information on to several groups that are enzymologists that are trying to understand the gene and how that works. But it is possible that it uses natural halogenated compounds as a supplemental electron acceptor.
Right.
Yeah. Because we isolate it from a pristine site, and we have seen it in many other sites that don't have. Well, there could be always some atmospheric fallout, but they're certainly not contaminated, heavily contaminated with fluorinated or with halogenated organics.
Yeah. So
makes me wonder about the role of enzymes in this whole thing, because they have created their own enzymes to break it down.
Yes.
Which is also a very specific and task to do.
Mm-hmm. Correct.
So what happens if you make the enzymes more? I don't know. Can you make them? Can you synthesize these enzymes, or do you need them from them to do things? Can you
well,
the enzymes?
I have a colleague that is trying to understand these enzymes and see if they can synthesize them, or maybe put the gene into another organism like an E. coli or so that grows much faster and produce them. That is sort of let's say a dream. And we don't know at this point. We know that there's one gene that is expressed every time we defluorinate.
Yeah.
PFAS. When we knock that gene out, there is no more defluorination. Although the organism grows as well as before, oxidizes ammonium at the same rate, but there's no defluorination. But I do not know if it is a single enzyme that does that it uses for the defluorination, or if it may be a whole train of different enzymes. If it's a single enzyme, then it shouldn't be difficult to synthesize it and maybe inject the enzyme and to treat the PFAS. But we don't know that if it's a more complex process or not.
Wow. Nature is so amazing. Oh my God.
Yeah.
So we're sort of like, we can do one question. It's 12 minutes for your hard stop, Peter.
Okay.
We can do one more question, one more deep dive. Mark, you have a question, or somebody from the audience? We have two people from outside. If you have a question, please post it in the chat. And otherwise, Mark is going to ask him another profound one.
Yeah.
If not, I have a whole document where I can still pick things out.
It's sort of amazing, Peter, that the bacteria you work on, these acidic microbiome A6, has brought you so many new insights and ideas, and it brings together two problems we have in the Netherlands, which is ammonia, or too high levels of nitrogen, and PFAS. We have both. We have too much of them, both.
Yeah.
And for some, Well, we've just begun to look at for acidic microbium in the Netherlands, and we
Right, but so far, I don't think there has been a direct hit.
No, not for A6, but we did
Yeah.
find acidic microbium. Yes.
Other acidic microbium, and there could be some. There are some sites also in Australia where they found in PFAS contaminated sites some very similar organisms to A6. They haven't shown that it defluorinates, but it's intriguing that it's there.
Yeah, but we really want to try to test the organisms that we find here in the Netherlands to see if they defluorinate. And we need some funding to do that, obviously. But I think, or maybe we find A6 in the end after all in other samples. I don't know.
Yeah.
Is it in your database, Mark?
Yeah, sure. We've been working together with Peter on finding those bacteria in the Netherlands, and well, progressively we got better at finding them. I have to say,
they're not true. They're tricky to work with.
Yeah. They're tricky to work with, and maybe are they somewhat sensitive to special conditions or?
Yes.
Yeah, right. So what do you think? What will what do you think is crucial to get this to work on a bigger scale to actually remediate the PFAS? You already said a little bit about it, but
well, on a bigger scale. So if you mean a contaminated site. There you have to get the redox conditions right, which is not so well. Environmental engineers and microbiologists have been successful for dechlorination to get the redox conditions right. Here, the jury is not out of the box. When I say that A6 has a hard time competing against heterotrophic iron reducers, we have to be very careful how we stimulate or change the redox conditions and not have Geobacter go wild and then push A6 out. The limitations and advantage is that A6 needs acidic conditions. The ideal pH is about 4.5, which is a pH that the typical heterotrophic iron reducer doesn't like. Geobacter doesn't do well, Shionella does not do well at that pH. So we would have to both bring the pH to and add some carbon to bring the oxygen down to get reducing conditions. That should work. Then there is, like I mentioned earlier, if you make the soil anaerobic to favor A6, after who knows, half a year, you will run out of ferric iron. So how do you renew the ferric iron? Either we have to supply it, which is challenging because it's a solid, or one could, of course, conceivably cycle the aquifer from oxic to anoxic, reoxidize iron, and we don't quite know yet how that will shock the system, how long it will take to restart.
So we have to cycle the iron actually. Yeah. Maybe other bacteria can help with that.
There are, of course, iron oxidizers, yes, but they need oxygen, right? So
they're nasty. They produce time and
Now, what is interesting? Just a few more minutes. We have studied the Neuse River sediments. The Neuse is in North Carolina. Those soils are acidic, and there's a lot of pork farming, and therefore a lot of ammonium that runs into Neuse. It's a very high ammonium level. So we decided to look if this organism is there, and we found it. And we finished our sampling scheme. And then Hurricane Floyd I think it was, I forgot the name of the hurricane, hit. And shortly thereafter, we went back and sampled. And what we noticed is that these big storms, due to the erosion they caused, they deposited a lot of ferric iron in the sediment. So the amount of ferric iron after that big hurricane went up substantially.
Up, and then they could grow again.
And then we could see that A6 was up by almost an order of magnitude. No? And that was sustained throughout the year. So it looks like there are natural cycling like that can boost it, and maybe one can do something like that in aquifers too.
Tidal areas where the water goes up and down.
Yeah.
We also have iron oxidized and reduced in one day cycle, maybe even.
Yes.
Yeah,
Oh, really?
that's absolutely super.
But that's interesting for water companies, or the ones who are managing the water levels in the Dutch polders.
Mhm.
That actually they have a big issue with CO2 emissions out of these low-lying lands. But if you use the concept, okay, but if there is iron in there and you can play around with it, then suddenly your whole bacteria flora will change, and then the whole soil will change accordingly, which is an interesting prospect.
Well, you certainly, it would have a bigger impact on methane emissions, right? Because if you reoxidize the iron, iron-reducing conditions are much more favorable than methanogenesis. So you could suppress methane.
Yeah, methane formation. Yeah, that's interesting.
We've done years back some modeling that showed that doing this cycling would, by reoxidizing iron, would suppress methane formation.
Actually, we are the western part of Holland consists of peat, and there's a lot of methane coming out of it, of course. And maybe it will get better if you would. Yeah.
Which is normally for mangrove, or let's say a type of low-lay land that's low. It needs the tidal to function.
It's interesting because the ecological functions are also a lot higher in areas where the water fluctuates more.
Yes.
Also true for bacteria, apparently. Yeah.
Yeah, that's a lesson we've learned today.
Lots of lessons to be learned.
Yeah,
today.
Peter, let's wrap up. Then you have time to transition to the another how do you call that? Beam me up, Scotty meeting.
Yes.
Hey, thanks so much for joining us on this podcast. It has been a privilege to dive deep in microbiology of topics I haven't heard of, but Mark is always the expert in that, and good to have a better understanding of PFAS and how microbes are able to crack it. Mark, you have an ending note?
No, it was a pleasure, Peter, to talk to you, and I really hope
it was fun. Yes.
Yeah, it was. I hope we will be able to work more, towards bioremediating PFAS in the Netherlands. I really hope we can do that. Yeah.
Okay, perfect.
I hope so too.
Yeah.
Then you can drop out, Peter, and all the best.
Thank you very much,
Bye.
Peter. Bye.
Pleasure. Bye.
I hope you enjoyed this episode of the Master Beautiful Achievements podcast. A special thanks to our sponsors Scope Matters and Orvion for making this series possible. If you like our podcast, subscribe in your favorite podcast app. And if you want to know more, do check out the mastersofbeautifulachievements.com website. If you like this podcast, do not forget to leave us a review on your preferred podcast platform.